Hi everyone, I'd like to thank you for the opportunity to present our work. My name is Marthe Collot and I am in charge of the production platform of the Imagene company. So we're going to present you our work on long-term room temperature storage of DNA through this talk called Storing Data Over Millennia. The Imagene company is an innovative company focused on ambient storage of biospecimen. We were created in 1999 in Bordeaux, where we still have our R&D lab, and now we have a biotech platform, a production platform based in a big European biopark in Évry Genopole near Paris. So at the beginning of the company, we were wondering how to store DNA for the long term. And you'll see from literature to industrialization how we managed to do it. The current storage approaches of DNA were in freezer and still are in freezers at -20 or -80 degrees. And this has major drawbacks. First of all, high costs of electricity, maintenance, air conditioning, alarm, etc. The second one is the difficulty in automating storage and retrieval of the samples. And the third one is also the risk of loss or degradation of precious sample collections or data due to technical failures like power outage or natural disaster. That's why we thought that room temperature storage would be very appealing, but how to do that? So we studied the literature about DNA, damaged sites, and the effects on the molecule. And we saw that a lot of sites could be attacked, and by different factors like water, oxygen, light, and ozone. And they could attack DNA on the link between base and sugar, on the phosphate, on the bases. And the main event is a loss of the base through depurination, and it's immediately followed by a chain break. And a lot of alterations of these different elements will lead ultimately to chain break. You could also have cross-links between bases and proteins and DNA, but these effects will strongly depend on the environment of the DNA. So from the literature, the conclusions are that the main and the most deleterious event is the depurination followed by a chain breakage, and that the main degradation factor is really water and oxygen, ozone, other atmospheric pollutants, contaminants, and co-extractants. They need water to act. And so that's why we thought that drying the DNA and keeping it dry throughout its lifetime would be the solution. That's why we tried at the beginning to find a suitable container that would be able to prevent a dehydrated molecule to be hydrated again. So we tried to find in the laboratories different types of vials, tubes, bottles, that could be hermetic. So we filled some of them with a water solvent, calcium chloride, that we weighed as a function of time. And we find out that no classical tube or vial is protecting DNA from moisture of the atmosphere. That's why we needed an hermetic container that we developed ourselves. It's the DNAshell mini capsule. So this is the technology that we use. So the stainless steel capsule is made of a metallic shell, a body, where we put a glass vial, and it's closed with a metallic cap. So after drying, the capsule is sealed by YAG laser welding, allowing to maintain the desiccated sample under an inert atmosphere. And for traceability, 2D barcodes are etched on the capsule, and the racks with 96 capsules are conform to SBS standard for automation. These capsules are quite small. They have a volume of 0.7 um, cube centimeters for usage volume of 300 microliters, and they weigh 1.3 grams. So this is the workflow that we use to put DNA in solution into the DNA shells. So the DNA in solution can be analyzed to have its quantity, concentration, the purity, etc. And it's aliquoted into the mini capsule with a liquid handler. The DNA in solution is desiccated with a primary desiccation into an evapoconcentrator under vacuum. We apply a low pressure and we heat the chamber so that the DNA doesn't freeze. And then after this first global removal of liquid water, we terminate the drying with the encapsulation station, which is a glove box containing argon and helium. And these gases are completely dehydrated, and we also remove completely the oxygen because we use molecular sieves to maintain this very pure atmosphere. And that's where the DNAshell is welded with the laser. And we get them out and we control the sealing with a mass spectrometer capable of measuring the helium that would leak from the capsule. Each capsule is measured. And so we have the DNAshell at the end, which is completely storable at any temperature, room temperature, and without no monitoring of moisture. So in our facility in Paris, these operations are managed through a laboratory information management system, and we have permanent tracking of the samples, the operations, and the mini capsules. And we can have up to 5,000 mini capsules in 8-hour shift of throughput. With this station called the BioShield Power Encapsulation Station, it's the higher throughput that we offer. So once it's encapsulated, the capsule can be stored and used in the different facilities. We can store them in a semi-automated storage station, completely at room temperature, with a high capacity of 250,000 mini capsules in a 3-meter square footprint. It's the equivalent of 10 - 80 freezers. We also developed the readers and the cherry pickers and the shell openers to be able to have a really easy usage of the capsule in the facilities. So talking about shell openers, this is how we recover the DNA. You open the mini capsule with this tool, one of these tools. In fact, you pierce the cap, you add water or buffer for the rehydration of DNA. This requires no other chemical steps. You briefly mix the solution by pipetting, and the DNA is ready for use. It's also automatable on a liquid handler. And we have a quantitative DNA recovery from picograms to milligrams, depending on the need of the customer. So the advantages of this technology is that we save energy. There is no energy consumption once the capsule is closed, no specific infrastructure, no air conditioning. Everything is easier at room temperature, the excess, but also the logistics, which become sustainable. We save costs because less space is required to store the capsules. The tracking is very easy with the 2D code that is etched, so it's really secure. And also, the capsule is tamper-proof. You cannot have no accidental opening or cross-contamination between capsules, because when it's open, you know it. You cannot reclose again a capsule without being noticed. And of course, the room temperature and the absence of all the degradation factors from water, oxygen, light is giving a strong integrity and stability to the molecules. And to prove that, we performed a lot of stability studies, and we published a huge amount of data to validate the technology. So at the beginning, the first thing we had to do is to know how to measure the chain breaking of the molecule. So we used a model molecule. It's a super cold plasmid, and this molecule is like this. It's a double helix, but it's super cold, and you just need one cut on one of the two strands of the DNA to have another form of this plasmid, which is called an open circle. And what is very interesting for the study, so it's very sensitive to chain break, and you can distinguish the two forms of the plasmids on a simple electrophoresis gel. That's how we managed to follow the supercoil content diminution through time to have access to the degradation constant, to the rate of degradation per second per nucleotide of DNA. And this is an exponential fit that can give us this value. So as we expected, very low degradation rates, we accelerated the degradation by heating. And so we performed stability studies at different temperatures so that we could have different degradation rates for each temperature. And that's how you can build an Arrhenius plot. So this is what we have. And we published this in Nucleic Acid Research. And it was the first Arrhenius plot for dehydrated DNA. So how to read this? So here you have the data point for each temperature. It's the degradation, the log of the degradation rate. And you see that you can extrapolate this to room temperature and have an idea of how stable the dehydrated DNA is. So here we obtained 1 to 40 cuts per 100,000 nucleotides per century. So dehydrated DNA is remarkably stable at room temperature. The second observation that we could make from this study is that the oxidation is enhanced by moisture. We measured the formation of 8-oxo DJ at different relative humidities from 75% to 1.4%. And so this is a marker for oxidation of the molecule. And we observed that that water is participating in this formation. And the third observation is that the storage of DNA in atmosphere is deleterious. We left DNA we dehydrated DNA and left it in the atmosphere for 7 years, and we measured its size, and it was 1.5 kilobase, and starting with a high molecular weight DNA at the beginning. So we calculated the degradation constant, and it was here. So you have a huge difference between degradation rates at room temperature for partially dehydrated DNA and completely dehydrated DNA. So our hypothesis here was that DNAshell would be able to protect DNA for very long periods of time at room temperature. And once the technology was fully developed, we were able to confirm this in a real-time study at room temperature. So in this study that we published in the year after, we compared different conditions. We dehydrated DNA and we left it in a closed tube, meaning it was exposed to the atmosphere, to the moisture of the atmosphere. And we have 3 different conditions that were exposed to the atmosphere. The first one is without additive. And after 1 month, it was not possible to analyze DNA because it was completely aggregated and absorbed on the tube. The second and the third conditions are the addition of a matrix. So in blue, you have the tree halos matrix, and in orange, a commercial matrix. And you can see that we can still measure the supercoid content, but the degradation was really very quick because no supercoid plasmid was left after these 8 months. And as a comparison, we also, of course, put the DNA, plasmid DNA, in the mini capsules without oxygen and water because it's laser sealed under argon, and also without any additive. And you see that even after a few years, the supercoid content is still very high. So with this study, we proved that the adverse effect of air exposure is really important for aggregation and degradation, and also that DNAshell affords an efficient protection and made it possible to have what we claimed before, meaning that if you have completely dehydrated DNA and if you keep it dry, it is very, very stable at room temperature. We recently benefited from an external validation of DNAshell for DNA data storage. And this study was performed by Organic and colleagues and by Microsoft Research, University of Washington, and ETH Zurich. They performed accelerated aging on 29,000 DNA sequences that they put in different storage conditions. And they performed qPCR and sequencing after rehydration. And they, for the kinetic, they used 65, 75, and 85 degrees. They compared these types of samples that were all dehydrated, and so in different conditions, without additive, with tree halos, with DNA stable, with a mix of sugars on a filter paper, with genetic raw matrix, and of course in the DNAshell here. And you can see it's quite obvious that we have a very important difference between all of these procedures and the DNAshells because of the absence of water and the fact that we maintain this dehydration throughout all of the lifetime of the sample. So from these, they could calculate the half-life of the fragments of 150 nucleotide long. And so no surprise to see that the half-life for the DNAshell is very high. So in this study, all of the conditions allow the qPCR and sequencing of the fragments, but you see that the DNAshell bring the best stability with this higher half-life. We are about to submit, we submitted, sorry, this new Arrhenius plot in the DNAshells. And so I will finish with this. We use this time not anymore the plasmid DNA supercoid content, but we use the two-size amplicon quantitative PCR system to determine the degradation rates at the different temperatures. Because we are aware that qPCR is really used for this kind of studies, and we wanted to show that there was no difference with the electrophoresis, and the measurement is also more precise. So the extrapolation with this Arrhenius plot here gave again 1 cut per century per 100,000 nucleotides. So we confirm here again that the DNAshell brings really high stability to DNA, and we can calculate from this that the half-life of 150 nucleotide long fragments at 25 degrees would be approximately of 40,000 years here. the best that could be calculated. And here we compared with the different other procedures, commercial or not, that we had access to. And some of them we measured them ourselves, and some are in the literature. So the first application of the DNAshells to DNA data storage was a UNICEF Norway initiative to celebrate the 30-year anniversary of the United Nations Convention on the Rights of the Child. The text was encoded by EMBL in UK. The DNA was synthesized by Swiss Bioscience, and we encapsulated the DNA. And now these capsules are stored in Norway. So in conclusion, the DNAshell brings really a technical superiority for DNA stability. This technology is now completely mature and extensively validated and also industrialized. It's already usable and used for DNA data storage, for example, for cold data and large amounts. Thank you for your attention. If you have any questions, please tell me.